Okay, ladies and gentlemen, in this set of notes, we're going to talk about DNA replication. Now, we've mentioned this before several times, particularly when it comes to something like mitosis. What happens during mitosis is you end up with two copies of a cell. And as we talked about, if you're going to get two copies, what that actually means is that you need two sets of chromosomes. And if you need two sets of chromosomes, that means that at some point you need a way to duplicate your DNA. Now, this process of duplicating DNA is what's known as replication. And so what we need to talk about is how this DNA is duplicated during the S phase of the life cycle of a cell. And that's going to be the focus of our presentation today. A couple things we need to talk about when it comes to DNA. First off, you hopefully at this point are familiar with the idea that DNA is this double helix structure. So what actually happens during the process of replication is the helix is separated. Okay? And if you recall, one side of the helix runs down, one side of the helix runs up. So the helix is separated. And then a new version of the helix is added to either side of it. Now, the reason this is so important is because no matter which side you have, you can use it to make the other side. That's why the complementary base pairing is so important when it comes to DNA. Because if I have the first side and the first side goes A, T, T, C, G, well, no matter what has to be on the other side is T, A, G, C. And so if I were to separate these and do T, A, G, C, and then put A, T, T, C, G, if I were to take this side and try to make a copy of it, it would look like this. And if I were to take this side and use it to make a copy, it would look like this. And so when you're done making copies of either half, they're both identical to the original that you started with. And that's the key functionality here when it comes to DNA replication. All right, the complementary base pairing is what allows duplication so easily. Now, to make this duplication happen, you need several different enzymes. If you recall, enzymes are proteins that cells use to speed up chemical reactions. So these enzymes actually cause chemical reactions to happen to the DNA, which allows the process of duplication to occur. We're going to focus on three enzymes right now. The first one is an enzyme known as helicase. Helicase unwinds the helix, allowing the other enzymes to actually get at each side so they can be copied. We generally use the term unzips, and helicase functions by breaking the hydrogen bonds between bases. Now, we also have another enzyme, and this enzyme is actually probably the most important in this process. And this enzyme is known as DNA polymerase. Okay? Now, DNA polymerase is an enzyme that has multiple jobs. For one thing, it adds complementary bases to a template. So back here, when I drew this example, and we talked about how if I have the blue template, I will make the green from it every time. And if I have the red template, I will make the green from it every time. and no matter which side I start with, I'll end up with the same thing as my original? Well, if you want to know how this happens, it's DNA polymerase. So DNA polymerase runs along this ATTCG strand and adds the complementary base. So next to A, it'll add T. Next to T, it'll add A. Next to T, it'll add A. And it would do the same thing at the bottom. It'll run along this red strand, and next to G, it'll add C. Next to C, it'll add G. Next to T, it'll add A. Okay, that's what DNA polymerase does. DNA polymerase is also brilliant because it is able to proofread. So DNA polymerase, while it's doing its job, also goes along the strand and makes sure that no bases have been added in the wrong place by accident. One thing about DNA is it only moves 5' to 3'. The carbons in the ribose sugar are numbered, as are the carbons in the nitrogenous base. And so what happens is, to distinguish the carbons in the sugar from the base, we refer to them as 1', which is what this apostrophe means, 2', 3', 4', and there's 5 of them. And when the phosphates connect the sugars to the backbone, they connect from the 5, they connect to the 5', to the 5', to the 5', to the 5', and they connect from the 3', from the 3'. So what this does is this gives DNA directionality. And this is actually what Crick was talking about when we watched that film, and he had the pencils. So one DNA strand runs up, meaning 5' at the bottom, 3' at the top, and the other DNA strand runs down, meaning 3' at the bottom and 5' at the top. They're just running in opposite directions, okay? It's like opposite sides of a highway. One side goes north, one side goes south, okay? And just the way we indicate that in DNA, we don't use north-south, we use 5' to 3', all right? The last enzyme that I want to talk about in this process is something called DNA ligase. DNA ligase is basically like a glue gun. It finds pieces of the DNA that have been made, so pieces of our new strand, and it connects them, creating one long strand. And you'll see why that matters in a second. Okay, so we got 3 main enzymes we're concerned with. Helicase, which unwinds the DNA helix. DNA polymerase, which adds the complementary bases, does some proofreading, and only can read from 5' to 3'. It's important to note that DNA polymerase is definitely the key enzyme here. And then we also have DNA ligase, which is sort of like our glue gun. It attaches unconnected pieces of DNA that have been newly made. So this is what DNA replication looks like. This is actually known as a replication fork. Okay, and this is the 5' end, this is the 3' end of one side of our double helix, which means this is the 5' end and this is the 3' end. Again, remember, north to south, south to north. They run in opposite directions. So this arrow right here represents DNA polymerase sort of just building a new strand. So do these arrows. They just do it in a little bit of a weird way. Right here is where I find helicase separating the strands so that I could get at this side and get at this side. This just shows us the direction replication is happening. So let's talk about this. This side up here is known as the leading strand. On the leading strand, things happen very simply. On the leading strand, DNA polymerase just reads, like the arrow says, from 5' to the 3' direction. And DNA polymerase sort of just follows helicase. And so what you end up with, as helicase pops open the DNA strand, polymerase follows along behind it, and you end up with here one new helix. Easy. Leading strand is very simple. All right. Down on the other side, however, things are a little more complicated. So this is what's known as the lagging strand. Okay. Now, the lagging strand is complicated because the lagging strand is backwards. So you have to remember, DNA polymerase can only read in one direction, just like you only read text left to right. And so to make this work, DNA polymerase will duplicate a section, jump backwards, duplicate another section, jump backwards. So it actually follows DNA helicase by jumping backwards. And what this does is this creates small chunks of replicated DNA. And we actually have a name for these small chunks. We call them Okazaki fragments. They're named after the scientist that identified how this half of DNA is copied. And this is great. I mean, like I've got my second DNA structure here, but they're not connected. And that's where DNA ligase comes in. DNA ligase connects the Okazaki fragments. And then what that does is, after DNA ligase is done doing its job, this creates another complete helix. When this whole process is done, I had one DNA helix, and my one DNA helix has been split in half. And then my DNA polymerase, my DNA ligase, my helicase, they've done one long leading strand. Then on the lagging strand, they did connections of the Okazaki fragments. And what that did is it created a situation where this first strand, which is half of the original, is now also half new. And this second strand, which is half of the original, is now also half new. A couple of odd things. This is known as semi-conservative replication. The reason we call it semi-conservative is because one half is old strand, one half is new strand. And the last thing I want to point out is the nucleotides that are added, you get those from your food. All right, when you break down other living things to digest them, your body takes those extra nucleotides. Or if you break down cells that got destroyed or are unhealthy or something, your body breaks down all the nucleotides to reuse them for this process of replication. All right, everybody, that's DNA replication.